So chapter 11, section 2 looks at the four functional components of the electron transport chain. So the first functional component is really the these three big multi-subunit protein complexes that traverse that inner mitochondrial membrane and some of them work to translocate protons across that membrane. Then the second functional component is coenzyme Q. So it's the green. I guess I shouldn't be in purple. I should be in green. Let's see, in green, right? And it's with and without a proton, but it's still coenzyme Q, which we call ubiquinone. That's where the Q comes from, in ubiquinone. It's a small hydrophobic electron carrier that diffuses laterally in that inner membrane, and it donates electrons to complex 3. So those electrons can come from couple of different places, but it will take it to complex 3. Then we have the 3 membrane associated enzymes. So these are the ones that have FAD inside of them. So you're kind of double dipping here, but it is complex 2. Complex 2, we talked about this. Complex 2, right, is that succinate dehydrogenase that's embedded in the inner mitochondrial membrane. And then one we're not going to touch on too much yet, and they don't have it on that drawing. And here they have it on this one. Well, so when we talk about fatty acids, right? We have this protein called ETFQ oxioreductase. And so this is only for fatty acids, and it takes 2 electrons from your fatty acid and sends it to the electron transport chain. And in the process, it makes acetyl-CoA. And we'll talk about that a lot more when we get to fatty acids. Then you could also get those electrons from glycerol 3-phosphate dehydrogenase. And it comes from the shuttle system, right? But this is our glycerol 3-phosphate dehydrogenase right here. And this is where we take it from the glycerol 3-phosphate and we make dihydroxyacetone phosphate. And we'll get more into those details. But all 3 of these, our complex 3, our ETFQ oxioreductase, and our glycerol 3-phosphate, they all pick up electrons, donate them to coenzyme Q, ubiquinone, which brings the electrons to complex 3. The fourth thing that we're going to talk about is fourth functional part of the electron transport chain is cytochrome C, which sits on the cytosolic side of the inner membrane. So it sits within the inner membrane space, and it carries the electrons one at a time. It's pretty particular about that. It can only carry 1 electron at a time from complex 3 to complex 4. So when we talk about this, let's kind of come back up here, right? We know that oxidation of NADH by the mitochondrial electron transport chain takes place on that matrix side of the inner mitochondrial membrane. So we're going to begin in our complex 1. Complex 1 is going to take those electrons from NADH, right? And the electrons from NADH, the 2 electrons from NADH, are going to cause a cascade of redox reactions. And it's going to go all the way through all of the electron transport chain, and it's going to end with oxygen, O2, being reduced to water. That's the very end. That's the culmination of more than 20 electron carriers, right? And remember that this happens in the form of a hydride ion. We talked about that before, that it's carrying 2 electrons, right? So we put the 2 little dots for the electrons, and because there's a proton and then 2 electrons, right, it carries that negative charge. So it's really 2 electrons and H plus, and that's what gives us our hydride ion. Okay, so the other way that we can have electrons enter the electron transport system is if they are in FADH2. FADH2 cannot drop off to complex 1. It must drop off at complex 2. And the reason is because of the standard reduction potentials. So each electron carrier has a unique standard reduction potential. And we're always going to go from high, right, we're going to go to a higher, a more positive standard reduction potential. So NADH is going to be the most negative, O2 is going to be the most positive, and our electrons flow in that direction. So you're always going to have that sequential arrangement of electron carriers. So a lot of times what you're going to see in the complexes is that you have proteins that have iron containing prosthetic groups, right? So you can have those different oxidation states and that kind of thing. All right, so when we talk about our electron transport systems, let me kind of erase what we were doing before. Kind of gets crazy. So you have to remember that we can have electrons come in at different places. So in this first example, our electrons are coming in from NADH. Where does NADH drop off electrons? It's always going to drop off to complex 1. We know that complex 1, once NADH drops off those 2 electrons, turns into NAD+, it's going to drop off 2 electrons. Those 2 electrons actually kind of ping pong around in here. And as they do that, they pump protons from the matrix into the inner membrane space. And so in complex 1, that's actually for every 1 NADH, you're going to pump 4 protons from the matrix side to the inner membrane space. Then if we look at what happens to those particular electrons, like we're trying to track the electrons here. So the electrons are going to get transferred from complex 1 onto coenzyme Q. Once Q picks it up, now we don't call it Q anymore, now we call it QH2. So QH2 is carrying those electrons, and those electrons get dropped off at complex 3. So notice how we skipped complex 2 here. So we have complex 1, which is our NADH, that's why we got it from it. So we call it NADH. We're going to give it to ubiquinone. So we call it NADH ubiquinone through oxidation reduction reaction. So it's oxidoreductase. Give it to Q. Q is going to take it to complex 3. We call complex 3 the ubiquinone because the electron started in ubiquinone. They're going to go through complex 3, and as they go through complex 3, they're going to pump an additional 2 protons, and then QH2 had 2 protons here, so you're going to get 2 from there for a total of 4 protons being pumped at complex 3. So once complex 3 has pumped those protons, we still have those electrons that came from Q. They're going to get separated in this really complex Q cycle that I will teach you. And one at a time, cytochrome C is going to pick it up. So ubiquinone is where the electrons came from. Cytochrome C is where they're going to. So you actually see that they're starting to name these things pretty well. So then cytochrome C is actually going to have to make 2 trips. So that's why they call it 2 times 1x. It makes 2 trips. And it drops off those electrons at complex 4. This is called cytochrome C oxidase because the electrons are done at this point. When those electrons get dropped off, they're going to be used to convert oxygen into water. And so that process, you're not dropping off the electrons anymore. They're in water. They're done from the electron transport chain. So we just call it cytochrome C oxidase because we're oxidizing cytochrome C. Hey. And in the process, we only pump 2 protons. And then eventually we'll talk about ATP synthase, but we're not showing that on here. Okay, so here's our alternate route. Where else could electrons enter the electron transport chain? Well, they can enter at complex 2 if they're in the form of FADH2. Well, where do we get FADH2? Well, if you remember back to the citrate cycle, if you had succinate and you convert succinate to fumarate, what enzyme does that? That's succinate dehydrogenase. And I generate FAD from it, FADH2 from it, excuse me. So that FADH2 is going to carry 2 electrons and it's going to get kind of bounced around, right? And those 2 electrons are going to get transferred to coenzyme Q, which we just abbreviate Q, but there are no protons that get pumped. Notice there's nothing saying, okay, we send 2 protons or 4 protons. Nothing gets pumped. It just gets transferred to Q, straight to Q. So we name it succinate dehydrogenase because its main function is really in the citrate cycle. And then Q is where we enter. So, succinate dehydrogenase has 2 names. It's succinate dehydrogenase and it's complex 2 of the electron transport chain. Okay, so remember how we kind of said Q can get its electron from a couple of different places. It can get those 2 electrons from complex 2. If we're talking about breaking down fatty acids to get energy, they're going to get broken down into FADH2 and acetyl-CoA. And using the ETFQ oxioreductase, those electrons that came from the fatty acids can get picked up by Q. Or in the shuttle system, which we're going to talk about later on, that shuttle system can take glycerol 3-phosphate, drop off electrons in the form of FADH2. So what do you notice that's common about all 3 of these? The electrons are coming from FADH2. They can get picked up by Q. So regardless of where the electrons came from, we convert Q to QH2. So QH2 goes to complex 3. And if you look, everything here is the same. So once you get to that point, the rest is the same. You just have to remember that you go, if you're in NADH, right? This is our NADH. This is our FADH2 pathway. So if we go from NADH, we go from 1 to Q to 3 to 4, right? So in the FADH2, we go from 2, and really it's 2 glycerol 3-phosphate dehydrogenase and ETFQ oxioreductase, but we'll talk about those in a little bit. You go from 2 to Q to 3 to 4. So the N pathway is very, very similar. All right, so when we talk about the electron transport chain and ATP synthesis, you have to remember that those 2 processes are linked, right? They're coupled reactions. So if we inhibit electron transfer in the electron transport chain, then the downstream things aren't going to happen either. So we're going to have loss of ATP synthesis. So if you look at some of the really potent things that will kill you are inhibitors that block the redox coupling. So rotenone is an example of one. It's isolated from plant roots and it's used in Amazon tribes. And they use it and they make poison arrows. Hydrogen cyanide. The hydrogen cyanide gas is what was used in concentration camp prison gas chambers. And the sodium cyanide's dropped into sulfuric acid. And then the cyanide binds to the heme group in a cytochrome in complex 4, and then it blocks the electron transport chain so that you can't reduce oxygen to make water, and no electron transport chain, you are going to die a very quick death. Carbon monoxide, we know we need oxygen in order for this whole process to go. So if your red blood cells are not carrying oxygen and instead are picking up carbon monoxide, because carbon monoxide has a tighter binding, then you know that you're going to block at complex 4, because complex 4 is exactly where we convert that oxygen to water. So it's important to kind of know the big things of where it's happening. Antimycin A is another one. It's an antibiotic that's produced by strep bacteria. And it's been used actually as a pesticide to prevent non-native fish populations from getting too crazy in different streams and lakes and things like that. So it's kind of cool. All right. So here's the important bit. And I've been saying this over and over and over again. So when we talk about standard reduction potentials, right? Generally, your reduction potentials will gradually increase with each reaction in the electron transport chain. So basically what's happening is, as we go down here, this is the pathway of the electron transport chain. That's not perfect, but in general it is. So, and you have to remember that all of these numbers that we're showing you here, these are done in the lab. And so standard conditions are not really in vivo, you know, in the cell, right? It could be different in the cell. And it's really, really hard to measure some of this stuff. So you have to remember that different concentrations, different environmental conditions can change the reduction potentials enough to basically make this work. Okay, so how is the energy that's released by redox reactions used to actually move protons, right? Because we need to move the protons from the matrix into the inner membrane space. And this is really kind of cool. I love being able to say this, is that we really don't quite understand it yet. But it's thought to include one of two different things. And it might be a combination of both, and we're just really not sure. So the first one is called a redox loop, where you actually have separation of protons and electrons on opposite sides of the membrane. And we're going to go into like way detail on this, and it's the Q cycle. And we'll talk about the Q cycle separately. And it's kind of detailed, so we'll kind of wait on that one until we get to it. The second one is a proton pump. So this one you're probably a little bit more familiar with, because it depends on protein conformational changes, right? We know that the pKa values of our amino acids, right? Those are functional groups. And when they change, when the protonation state changes, we know that we can have an induced conformational change. So this is what we think is happening in 1 and 4, where 1 is the NADH ubiquinone oxidoreductase, and then 4 is the cytochrome C oxidase. And so what actually happens is that those two protons are transferred, right? So we have XH2 here, and it's going to transfer those two protons to this other carrier that's embedded in the membrane, right? And that is going to form a complex with those two protons, and at the same time also the electrons, and it's going to transfer them to the next complex. And at that next complex, because of that transfer, it's going to induce a conformational change, and then that conformational change is going to allow those two protons to be pumped. So it all depends on protein conformational change. The Q cycle is very different from that, but we'll talk about that in detail later. So let's kind of go through these different complexes one at a time. They all have prosthetic groups. So remember, go back and remember what's a prosthetic group, like a vitamin, a lipid, a metal ion, not an amino acid, that functions as electron carriers. So the first one, and really the largest one, it's got 50 to 40 polypeptide chains. That's like really, really big. So we call this NADH ubiquinone oxioreductase. So where do the electrons initially come from? They come from NADH. So where did the NADH come from? What's one place that it could come from? It could come from the citrate cycle. It's just one of the places. So we're going to take that NADH and we're going to oxidize it. And at the same time, coenzyme Q is going to be reduced. Remember, oxidation and redox, they're coupled. So it has a covalently bound flavin mononucleotide. FMN is very, very similar in structure to FAD. So like, you know, it accepts its electrons. So it's another electron carrier. So remember your oxidation and reduction. If you have to go back and do oil rig, that's okay. So oxidation, we're going to lose our hydrogen, and reduction, we're going to gain it. So if you look at the different components, NADH is being oxidized, Q is being reduced. So make sure you get that really, really solidified in your brain. Okay, so when we look at complex 1 structure, we have to remember that those electrons are flowing between multiple carriers. So remember how I told you it was kind of like ping-ponging around? This is what I'm talking about. So if you're in the matrix, right? And not the movie. When you're in the mitochondrial matrix, you have oxidation of NADH. So this is our very first step. We have NADH. I've been already drawn on this. We have NADH. Our NADH gets oxidized into NAD+. That is going to release 2 electrons. Now, here's the thing. What form are those 2 electrons in, right? Remember we talked about this. This is where we're going to transfer them to the FMN, which is really, really similar. And so we go through that hydride ion. So we're going to take those 2 electrons. They're going to start at FMN, and then they're going to get bumped to iron sulfur complexes. And they keep going to iron sulfur complexes. And they just keep transferring and transferring and transferring until we get to Q, right? So this iron sulfur, it's actually like 7 iron sulfur centers that actually have to do it, and they carry 1 electron at a time, and then they couple them. And remember that iron can change its oxidation state, right? So we can reduce it going from the +3 to the +2. But it varies in reduction potential based on the local environment. Remember, we always want, as we're going this way, we always want to see an increase in our reduction potential, right? So just kind of keep that in mind. Okay, so we've bounced around our 2 electrons through our iron sulfur centers all the way until we get to Q. Q is going to accept the 2 electrons, so 2 electrons, and here's the tricky part, it's going to accept 2 protons from the matrix. So we need both the 2 protons from the matrix and the 2 electrons from complex 1. So that is going to convert Q, ubiquinone, so this is O-N-E, I believe, ubiquinone, into ubiquinol. So ubiquinol has both the electrons and the protons. Okay, that whole process, all the way from dropping off those electrons to making ubiquinol, is going to transfer 4 protons from the matrix to the inner membrane space. So this is why we call that a proton pump. Proton pump, right? So the net result is that we've oxidized NADH. We've reduced coenzyme Q ubiquinone into QH2 ubiquinol. We've translocated 4 protons from the matrix to the intermembrane space, right? So those are 3 big roles in complex 1. So if we look at kind of the structure of our FMN reactions, you're going to see the same nitrogen containing rings and things like that. And they pick them up one at a time in order to make those semiquinone. This is your semiquinone intermediate, right? To all the way your reduced flavin mononucleotide. But you don't have to remember structure. That's fine. Okay, so coenzyme Q, that's where we kind of left the story off. Coenzyme Q has 3 really, really big roles, and it acts first as a mobile electron carrier, and it transports those electrons from complex 1 to complex 3. It's not the only place that it gets electrons from, but it's the one we're going to talk about in detail right now. It's also the entry point, so this is kind of 1. It's also the entry point of 2 electrons from multiple places. So from the citrate cycle, from fatty acid oxidation, and from, this is our shuttle system. Yeah, shuttle system of glycerol 3-phosphate dehydrogenase. And we'll talk about that kind of later. And then it converts those 2 electron transport system, right? Where we were in complexes 1 and 2, we had 2 electrons moving at a time, to the 1 electron that cytochrome C needs. And the way that those 2 electrons are separated into 1 electron at a time is through the Q cycle. And I have a really nice video in the modules, in like the exam 1 module, that goes through the Q cycle if you want like lots and lots of detail. So we've talked about ubiquinone. It's being reduced. When we go from Q to QH2, that's a reduction event. And as that happens, our 4 protons are again translocated. So if we look back here, not here, well, they must have one down here. I keep jumping ahead of myself, right? Okay. I went too far. Here we go. So, yeah, it doesn't really show it too well there. So if we look, we have 2, here's our 4 protons that are coming in. Here's our 4 protons. So where do we get the 4 protons? We're getting 2 of them from the QH2, and we're getting 2 that are pulled in from the matrix. So that's where our 4 protons come from. So just kind of keep that in mind. Okay. So when we talk about our coenzyme Q, have you ever heard about taking CoQ10, right? So if you look at the hydrocarbon tail of coenzyme Q, they're just putting it here in brackets, right? So if you look in, I kind of covered it with my mark, right? But if you look in humans have a 10 carbon isoprenoid unit. So we have 10 of these. I keep switching to race. We have 10 of these isoprenoid units in a row. And so that's why they call it CoQ10, if you've ever seen that supplement, that dietary supplement. Yeah, that's kind of why. So it picks up one at a time, right? Semiquinone to a reduced. Okay. So now let's talk about complex 2. Complex 2 is another big multi-subunit membrane-bound enzyme. We also call it succinate dehydrogenase. And so we know from the citrate cycle that we are able to take succinate and we're able to convert that succinate into fumarate, and in the process, we're going to generate FADH2, right? Now, FADH2 is my electron carrier, and it's going to drop it off, those electrons, it's going to drop off those electrons to iron sulfur center. It's actually 3 iron sulfur centers, and a B-type heme. So this is a B-type heme. And it's in the cytochrome, ooh, the cytochrome B560. Like, remember how we've said cytochrome C? There are different versions of cytochrome. And so this is cytochrome B. That's all it is. Okay. So it's the same thing where we have coupled redox reactions, but in this case, we're using our FADH2 and FAD. And in the end, what do we want to do with those electrons? Well, we want to give them to Q. Just like we did in complex 1, we again want to give those electrons to Q. So what you're not seeing here is there are no protons being pumped, but just remember that Q has to pick up its 2 electrons and it has to pick up 2 protons in order to turn into QH2. So just kind of keep that in mind. Okay, so now QH2 is going to take those electrons to complex 3. So Q ubiquinone, it's actually QH2, right? It's going Q to QH2, is going to drop those electrons off to cytochrome C, right? So this is why we call it ubiquinone cytochrome C oxidoreductase. So complex 3 is really a dimeric complex of like 22 protein subunits. It's a lot. And it reduces cytochrome C, and it does translocate those 4 protons. So QH2 is going to dock on this enzyme, and cytochrome C is going to dock on this enzyme. And that's what's going to allow those electrons to be transferred from QH2 to cytochrome C. So what's kind of interesting is that this complex has to have 2 binding sites. You put 2 in there, 2 binding sites for ubiquinone. It's QP, and remember that P is the positive, that's what it stands for. It's IT, positive, which is that intermembrane space. And QN is for the negative, and the negative is the matrix area. So it's going to transfer 1 electron at a time, and it's going to use this time iron sulfur complexes. So this is, remember, this is 1 electron. So this is 1 at a time, right? So we're going to have like QH2, and it's going to have 2 protons from the negative space plus 2 cytochrome C. And remember that those cytochrome C's are oxidized, in parentheses. And what are we going to make, right? We're going to drop off our electrons and our protons. We're going to end up with Q. We're going to end up with 4 protons that are now on the positive side, or the intermembrane space. And we're going to end up with 2 cytochrome C's, but now they're going to be in the reduced state. So just kind of keep in mind what's happening in order to make that work. Okay. Why do I already have writing? Oh, I think I came and talked about this. Okay. So complex 3. Our complex 3, remember, this is our ubiquinone cytochrome C oxidoreductase, right? So get comfortable kind of talking about this. And this is where we're going to kind of talk about the Q cycle. So when we talk about that, you have to remember what's happening, right? What's coming in is going to be QH2. Now, to make all of this balance, we're going to show 2 QH2s coming in. So you just got to kind of keep that in mind. So in that initial step, what's going to happen is we're going to have oxidation of QH2. So what happens when QH2 is oxidized? One of those electrons, right? Because because QH2 is carrying 2 electrons. In order to get Q, what happens is we need to transfer 2 electrons, but those 2 electrons need to be split up. So one of those electrons is going to go to an iron sulfur center that is eventually going to take it to cytochrome C, because cytochrome C can only carry 1 electron at a time. The other electron is going to be picked up by a heme. Remember we talked about that was heme. And those electrons let's go back. So then over here, our Q here, this is our oxidized Q. It's in the inner membrane space area. That's the QP. So we call this the QP site. The Q that's in the QP site is going to move after it drops off its 2 electrons. It moves to the QN site. Once it's in the QN site, it stimulates the electron to be transferred from the heme group, right, to the Q at the QN site. So that gets converted into the semiquinone, right, because it only picks up 1. Then a second QH2 has to go through that whole process. Go to the QP site, release its 2 electrons, 1 goes to the heme down this way, 1 goes up to the cytochrome C. Now I have another electron, and that semiquinone can pick up that second electron and turn into QH2. So this is kind of a hard thing to keep in mind, that you have that semiquinone sitting in the QN site until it picks up a second electron, right? And so then we regenerate QH2. Now, that second Q that dropped off its electron, it's in the Q form. So that Q gets released, it goes back and picks up more electrons at complex 2. So you just kind of have to remember that this 2 here really means that you have to go through the Q cycle kind of like twice. It's kind of weird. Okay. And we already talked about all that. Yeah. So when we look at cytochromes, so remember we taken our electrons and we're going from QH2, we're going through complex 3, which is the ubiquinone cytochrome C oxidoreductase. And we're going to give those electrons one at a time to our cytochromes. So our cytochromes, you can have different variations, right? So cytochrome C, which is what we are talking about in the electron transport chain, cytochrome C is a small protein and it associates with the inner membrane side, but it's soluble, so it can be in the inner membrane space, but we say it associates with the inner membrane side. And this is the one that's responsible for transferring that 1 electron. So you get 1 electron that goes from complex 3 to complex 4. And it's an iron containing heme prosthetic group that we've seen before in hemoglobin. It's a very highly conserved system. So remember that there are other types of cytochromes, and we've talked about them. Remember when we were saying the B type? Those are all examples of membrane bound. So the B type is actually in complex 2 and 3, whereas complex 4 is the type A. So that's really the type A. And these are all both membrane bound in the electron transport chain. So, and just remember that all you're doing is converting that iron from a 3+ to a 2+. That's all you're doing. Okay. So now complex 4, we're almost there. Cytochrome C oxidase. So we've got that electron, it's in cytochrome C, and we've had to do this twice. So those 2 electrons that come from cytochrome C making 2 trips is going to drop off electrons. And it's going to go through 2 copper centers. It's going to go through 2 heme groups. And eventually, right, we're going to transfer those electrons to oxygen. So as that's happening, you are going to actually pick up 4 protons from the inner membrane space. 2 of them are going to be used to be pumped. 2 of them are going to be used in converting O2 to water. So what you really have to kind of keep track of is our protons and our electrons. So we're only going to pump our 2 protons out, because remember, complex 4 only pumps 2. And then if we look at our reaction here, this is a really, really weird reaction. When have you ever seen a coefficient that's 1/2? And the reason that we do this is really you need 4 electrons in order to reduce 1 O2, to reduce 1 molecule of O2. So that's why we have that 1/2 there, just because you have to go through all of this twice, right? So now we have our final electron acceptor, right, is O2, and it turns it into water, and the electrons are done, right? So once the electrons are done, now we kind of have to summarize what we've done and see where we are. So we know that we have, I'm going to kind of ramble, I guess. We know that we have really 3 key elements that we've talked about so far, right? We've talked about complexes 1 through 4, right? Those all contain those transmembrane regions, the functional domains that face the matrix. But 3 and 4 need to transport electrons via cytochrome C, so their functional domains actually also extend to the inner membrane space. So you got to be a little bit careful about that, right? Because we need this part of it to face that inner membrane space. Okay, and we've oxidized if we have NADH, right? So if you start with NADH, that's going to complex 1, and that results in 10 protons that are being moved from the negative area to the positive area, or from the matrix to the inner membrane space, right? On the other hand, if we talk about, so this could kind of be, I don't know, the same sort of deal, but if you had electrons that were in FAD, H2, and really Q, right, you're not going to get all 10, because you're skipping complex 1. You're actually skipping complex 2 if you're starting at Q. So you're really only going to get 6 protons that go from N to P. So go back and look at those figures so that you start to see those differences between NADH and FADH2. Okay, and then we also talked about coenzyme Q, and we talked about cytochrome C, these being our electron transporters. Some are embedded in the membrane, some are free flowing. And remember, Q takes 2 electrons at a time. Cytochrome C can only take 1 at a time. So the next thing that we really kind of have to talk about is how efficiently the electron transport chain actually does its job. How well does it convert redox energy into a proton motive force? And so if you go back and remember our delta G naught prime. I know this is what you were missing so much of a redox reaction. We can actually calculate this from a standard reduction potential. So like I'm gonna kind of draw some things on the side. Okay, so if we have delta G naught prime, we know that this is our formula. Okay, so we have the N is our number of electrons, right? So if we have NADH, that gives us 2 electrons. F is Faraday's constant. Faradayy, I think. Faraday's constant, which is 96.48 kilojoules per volt mole. Hello, per volt mole. Okay. And then our delta E naught prime, this is from our table in the book. I think it's the very first table in chapter 11. So it should be table 11.1, right? And so how do we calculate this? How do we figure this out? Well, we know that comes from the difference between the acceptor and the donor, right? And so we're always going to say the acceptor, not prime, to the donor, right? And so if you look at, for example, let's compare our two initial electron carrier and final. So if you look at that, our final electron acceptor. So if we look at that and we plug those values in, so we would have to do this for the 1/2 O2. Remember that, that's not weird, but so this is E naught prime, and this is going to be NADH. So if you go and you look up those values, and I think I've had them on the earlier slides, right? So your O2 is a positive 0.8, I think 2 or 3. Okay, and then your NADH was really negative, right? Negative 0.32 volts. So if you look at this, this is a difference. This is a huge difference of positive 1.4 volts, right? So now if we take this and we plug this in to our delta G formula, right? So our delta G is equal to negative NF E of whatever the cell is, whatever, you know, the system is, and in this case it's the cell. So this would be delta G not prime, ha ha ha. Okay, so that would give you negative 2,196.48 kilojoules per volt mole times now Faraday's, oh, why did I do that? Wow, it's -2. Why did I do that? -2 is our number of electrons. 96.48 is our Faraday's constant. And then E of the cell, we're going to get from right here, and we're going to plug it in, 1.14 volts. So if we do the math on this, delta G naught prime gives us negative 220 kilojoules per mole. That's theoretical. It's theoretical, but this is the amount of free energy we have per 2 electrons from one NADH. That's amazing. I need more room. I'm already running out of room. Okay, so it's theoretical. It's not actual. We actually have to take into account the difference in pH, the charge, the membrane potential, the temperature, all of that kind of stuff. So if we do that, we're going to use this formula up here, right? So if we go ahead and plug in what we know into this formula, so delta G, R, we know what R is, right? R is our constant, 8.314 times 10 to the -3, because I want this in kilojoules per mole K, right? Then T, what's our temperature? Well, if we're in the human body, we got to be at 37 degrees Celsius, and 37 degrees Celsius is really 310 Kelvin. Okay, then we're going to do the natural log of our concentration. Now this you have to be given, right? So I know, I'm going to give you this. You don't have to know this off the top of your head. But if you have a change in pH from one side of a membrane to the other, right? I know that difference is about 0.9 in the mitochondria. So if you were to look at the ratio of concentration of protons on one side of the membrane to the other side of the membrane. So it'd be C2 over C1. That's about 1/8, right? Which gives you 0.125, right? So when you look at that, you don't have to know this. You would just have to be able to use this. So don't freak out. Okay. So now I have a value to plug in for my ratio of my concentration of protons on one side of the membrane versus the other side. So this is 0.125, right? Okay, then plus, we keep going. All right. So now Z is going to be our charge, right? So when we look at our charge of the molecule that needs to move, that's our proton. What's the charge of a proton? It's +1. Then Faraday's constant, we said 96.48 kilojoules per volt mole. Why do I keep getting that wrong? And the last thing we need is our membrane potential. So you would have to be given this. And I'm telling you that the membrane potential due to a pH difference of 0.9 would be negative 0.16 volts. So if I put all that together, right, what are you going to get? So do all the math, right? And what I kind of want to do is I want you to separate this factor from this one. So don't completely simplify it yet. So you should get a delta G that our first kind of unit there is negative 5.36 kilojoules per mole, plus we have a negative 15.44 kilojoules per mole. So when we look at this, right, our total delta G is going to be negative 20.80 kilojoules per mole this is really per proton, right? Or really per NADH, right? However you want to think about that. But when we look at how many protons are we going to translocate, we're going to be translocating 10. So we want to multiply this. I'm so running out of room. My goodness. We're going to multiply this times 10. So you're going to get about negative 208, right? Kilojoules per mole. Don't look at my sig figs. So of this, negative 208 kilojoules per mole of energy that we have, where does all of that free energy come from? Does it come more from our change in pH, right? That's our concentration on one side versus our concentration on the other. Or does it really come from the membrane potential? And what we can see is that way more of it comes from the membrane potential than comes from our change in pH. So that's kind of an interesting thing to look at. And we said, okay, what was our theoretical? Our theoretical down here, let's see, our theoretical was 220 and our actual was 208. Gosh, that's pretty good. What is that, about 90, 94, 95% effective? So what happened to the difference? It got lost as heat, right? So in the next section, we're going to see how free energy gets really converted into ATP. So make sure you can kind of summarize for yourself what's the role of coenzyme Q in the electron transport chain, just different things like that. Be able to summarize for yourself what was important, that kind of thing. All right, and I'll see y'all on Thursday.